Flow state, often described as being in the zone, is a psychological and neurophysiological condition characterized by effortless concentration, a diminished sense of self-consciousness, and a heightened capacity for sustained cognitive work. For students, flow can transform study sessions from fragmented, shallow review into deep, efficient learning episodes that enhance comprehension, retention, and problem-solving. Understanding the neurochemical and network-level mechanisms that underpin flow clarifies why it facilitates learning and suggests practical, evidence-based strategies students can use to cultivate it. Neurochemical foundations of flow. Flow emerges from a coordinated interplay among several neuromodulatory systems that shape attention, motivation, mood, and reward processing. Three classes of neuromodulators, dopamine, serotonin, and endorphins, play central and complementary roles. Dopamine drives goal-directed behavior, reinforcement learning, and the allocation of cognitive resources. Phasic bursts of dopamine signal prediction errors and salient feedback, sharpening attention to task-relevant cues and reinforcing patterns of action that lead to progress. During flow, an optimal balance of tonic and phasic dopamine promotes sustained engagement and the subjective sense of effortless control. Insufficient dopamine reduces motivation and working memory updating. Excessive, noisy dopamine can fragment attention or promote impulsivity. Serotonin modulates mood, behavioral flexibility, and the filtering of distracting signals. It contributes to emotional steadiness and the tolerance of challenge, both essential for persisting with demanding study tasks. Serotonergic tone influences the balance between exploring new strategies and exploiting known effective ones. During flow, serotonin helps maintain cognitive focus while permitting appropriate adaptive adjustments without derailing concentration. Endorphins and opioid peptides, endogenous opioids, endorphins, enkephalins, contribute to reward, pain modulation, and affective regulation. Their release can reduce the perception of cognitive fatigue and stress, facilitating prolonged high-effort engagement. Coupled with dopamine's reward signaling, endorphins help sustain the positive hedonic quality of flow that makes deep study intrinsically reinforcing. Network dynamics and cognitive control. Beyond neurochemistry, flow reflects emergent patterns of large-scale brain network activity. Two networks are particularly relevant. The task-positive network, including dorsal attention and frontoparietal control regions, supports sustained attention, working memory, and goal-directed control. In flow, these regions exhibit focused, efficient activation enabling effective processing of task demands. The default mode network, DMN, associated with mind-wandering and self-referential thought, typically deactivates during flow. Reduced DMN engagement minimizes distracting internal narratives and performance anxiety, allowing uninterrupted concentration. Efficient flow also depends on functional connectivity between the prefrontal cortex, PFC, and subcortical structures, including the striatum, mediating motivation and action selection. Optimal PFC modulation, neither hyperactive self-monitoring nor under-controlled distraction, creates a sweet spot where cognitive control is present but automaticity increases, supporting both accuracy and speed in learning tasks. Conditions that foster flow in studying. Flow emerges when task demands align with skill level and when clear goals and immediate feedback are present. Translating this into study practice involves managing both internal states and external environment. Clear, proximal goals and immediate feedback. Break study material into narrowly defined objectives, e.g., solve 5 calculus problems targeting one technique, summarize a single article's argument. Use practice problems, self-testing, or instant feedback tools, flashcards with answer checks, practice quizzes, to generate actionable performance feedback and small, frequent dopamine reinforcements. Optimal challenge skill balance: select tasks slightly above current competence to induce engagement without frustrative overload. Gradual scaffolding, progressively increasing difficulty, maintains the sense of forward momentum required for flow. Minimize distractions and encourage sustained attention. Create dedicated, low-interruption study blocks, commonly 50 to 90 minutes depending on individual capacity, with a single, prioritized objective. Use environment management techniques: phone on Do Not Disturb or in another room, browser blockers for distracting websites, and a tidy, well-lit workspace to reduce extraneous cognitive load. Regulate arousal and mood. Brief physical activity, 5 to 15 minutes of aerobic movement, elevates dopamine and endorphins and primes attention networks. Moderate caffeine intake can transiently enhance dopaminergic signaling and alertness but should be titrated to avoid jitteriness that disrupts focus. Practices that reduce anxiety, deep breathing, short mindfulness exercises, or progressive muscle relaxation, can modulate serotonergic pathways and reduce DMN intrusion, easing entry into flow. Structure for intrinsic reward and meaningfulness: connect material to personal goals or curiosity to strengthen intrinsic motivation. Perceived value amplifies dopaminergic reward signals, making extended effort more sustainable. Gamify study sessions, time challenges, point systems, to create additional immediate feedback and reinforcement. Leverage spacing, retrieval, and varied practice. Flow on a single focused task benefits encoding, but long-term retention also requires spacing and retrieval practice. Alternate deep flow sessions on discrete topics with interleaved review sessions that encourage retrieval, which reinforce neural consolidation beyond the immediate flow episode. Practical session design: a template pre-session, 5 to 10 minutes, brief aerobic warm-up or mobility, set one clear study goal, and remove distractions. Flow block, 50 to 90 minutes. Focus on a single challenging task with periodic micro-checks, self-testing every 15 to 25 minutes, avoid multitasking. Short break, 10 to 15 minutes, relaxation, light movement, or social interaction to allow endogenous reward systems to recalibrate. Consolidation, 10 to 20 minutes, retrieval practice or summarization to strengthen encoding. Individual differences and limitations. Neurochemical baselines and network dynamics vary between individuals and across contexts: sleep deprivation, stress, psychiatric conditions. For example, ADHD involves altered dopaminergic functioning that makes sustaining flow more difficult. Medication, structured environments, and tailored task segmentation can help. Excessive reliance on stimulants or poor sleep harms the neurochemical balance necessary for authentic flow and durable learning. Finally, while creating conditions for flow is valuable, alternating deep focus with passive rest and varied practice remains essential for memory consolidation and creative insight. In conclusion, flow state represents a confluence of neurochemical modulation and optimized network dynamics that together produce concentrated, efficient learning. Dopamine, serotonin, and endorphins each contribute distinct but interlocking influences: motivational drive, mood and cognitive flexibility, and effort-related hedonic buffering, that enable prolonged, effective study when environmental and task conditions support them. By structuring study sessions with clear goals, calibrated difficulty, immediate feedback, distraction control, and arousal regulation, students can routinely enter and sustain flow episodes, maximizing comprehension, retention, and academic performance while preserving cognitive health.